I want to talk about is our view of HIV, which is a virus that infects people, and it's actually, thank you, a very devastating virus that would actually, you know, if you think about it as a war between your immune system and HIV, HIV is definitely going to win. It's very highly evolved to go after all kinds of things in your immune system so that your immune system can't work properly. So in this country, as I'll talk about, we have antiretroviral drugs, so it's not so much of a problem here, but it's definitely a problem worldwide. So about 35 million people in the world today are currently infected with HIV. Not all of them have AIDS, of course, but they're infected, and if they don't get antiretroviral drug treatment, they will eventually progress to AIDS, and then people die. I mean, you don't live with having AIDS. You can for a while, but most, the vast majority of people eventually die. And this is a problem not so much in the United States and Western Europe, it's a problem in sub-Saharan Africa, for example. And there are places in sub-Saharan Africa, especially South Africa, where, for example, 67% of women who go to a clinic for pregnancy tests are HIV infected already. So it's not working to take the drugs that we have and distribute it all over the world. There's a lot of sociology reasons and things like that, but the point is that this is a huge problem worldwide, and over half of these 35 million people who are infected are women. It's not a gay male disease the way we think of it in the US, for example. So I want to talk about HIV itself so that we're all on the same page with what this is. So it is a virus, and like other viruses, for example, poliovirus or common cold virus, it's what we call an enveloped virus, and that means that the outside of it has a lipid bilayer membrane. That's like the cell membrane around all your cells. It actually acquires it from your cell when it's inside your cell. So it has a lipid bilayer that's derived from the host cell, and we're going to be talking a lot about these spikes that stick out of the virus, and I will probably lapse into terminology where I refer to the spikes by their names that we call them, which are GP120 and GP41. That has to do with their molecular weights. So these are just the way we refer to it. But what I want to show you now is how HIV works in sort of a summary of this. Okay, so basically, the purpose of it and any other virus is to get its genetic material into a cell so that then the cell will do the work for it, because the virus itself is a parasite. It doesn't have the capacity to reproduce on its own. It uses stuff in the host cell. So what it wants to do is put its genetic material, which in this case is RNA, into a cell, and then have all the cell machinery make more viruses. And then those viruses get out, and then they infect more cells. So that's the purpose of any virus, and in particular, we're talking about HIV. So then I wanted to highlight the lipid membrane here. What we're talking about is this thing on the outside. It's just a bilayer, if you remember, that surrounds the cells in your body or in, animal cells or whatever. And then I'm going to be talking about these, what I'll call envelope trimers, and that means proteins that stick through the membrane bilayer, and I'll call them either spikes or envelope trimers. That just means there's 3 subunits, and the subunits are called, as I said before, GP120 and GP41, okay? So that's kind of the definitions. Okay, so here's what is happening. It's a quick summary of the HIV life cycle. So basically, here's the cell, it's the host cell, and it gets, here's a virus right here, and the spike trimers or envelope trimers on that virus bind to the host cell. And so how does it bind? Well, the GP120 part of this spike trimer binds to the host receptor, CD4. And we're going to come back to CD4 in a moment, because this is critical for why HIV is such a problem to us. But anyway, just to go through this then quickly, It gets into the cell, and I'll talk a little bit about how it does that, but basically the lipid bilayer of the virus fuses with the lipid bilayer of the cell, because the cell has the same type of lipid bilayer, and they fuse. And that's one of the things we're studying in my lab. And the genetic material of the virus, which is RNA, gets into the cell, and then it integrates into the nucleus, and more viruses eventually get made, and they bud out of the cell, and that's why they have a lipid bilayers. They take it from the host cell. Okay. All right, so I said I was going to talk more about CD4 here. That's just a protein on the surface of certain cells in your body. And the fact that HIV uses CD4 as its receptor is one of the reasons that HIV is a problem for us, because CD4 is a molecule that's on your immune cells. Okay, so HIV is going to infect CD4 T cells. So you may have heard about T cell counts and stuff going down in HIV infection. So what happens in an untreated HIV infection is you would normally have a certain number of CD4 cells, let's say 1,000 per microliter or whatever. And then if you get infected with HIV, in the first couple of weeks, your CD4 count goes way down. And the person would have something that felt to them, they'd have a fever, they'd feel like they had the flu, but they wouldn't be deathly ill. And then eventually their CD4 count would go back up. Never high, quite high enough, but it would go back up. But over the period of, let's say, the next 10 years, their CD4 count would go lower and lower and lower. And when it gets below about 200 per microliter, starting with 1,000, the person is going to get all kinds of opportunistic infections. And I'll explain that in a moment. But the point is that these CD4 T cells are a type of white blood cell, and they control other immune cells. And so when HIV infection results in low levels of CD4 cells, you don't have a functioning immune system, and you're going to get what are called opportunistic infections. So these are, things that are in your environment that just normally your immune system, you're exposed to these all the time, but you have a functioning immune system, so it's not a problem. But if you don't, there are these things in your environment, for example, various species of yeast, which would give you thrush, or TB, the bacterium that gives you tuberculosis. You can activate latent varicella zoster, that's the chickenpox virus that would lead to shingles. Many people are infected with Epstein-Barr virus if you've had mono, and then you get B-cell cancers right here. There's a type of herpes virus that leads to, when you hear people talk about Kaposi sarcoma, where people get the purple spots who have untreated HIV infections. There's a particular type of fungus that can lead to pneumonia. A lot of people are co-infected with hepatitis C, and so on. And there's all these opportunistic infections that happens, and the person eventually dies, not from the HIV infection, but from the huge number of opportunistic infections. Okay, so that's what happens if you're not treated. Now, I also talked about how the genetic material of HIV is RNA. But actually, HIV is part of a family of viruses called a retrovirus. And what a retrovirus is something that takes its RNA and makes it into DNA. And this is, if you remember from biology, DNA goes to RNA, goes to protein. That's called the central dogma. Well, for a long time, people didn't think it was possible to reverse transcribe, that is, go from RNA to DNA. But it turns out it is possible, and you can do it. Viruses do it. We actually do it, but this is like discoveries 30, 40 years ago. But anyway, the central dogma is not true because you can go from RNA to DNA. So because it's not common to do that in human cells, the virus carries its own enzyme to reverse transcribe, and that's called, cleverly enough, reverse transcriptase. And reverse transcriptase, the HIV reverse transcriptase, is amazingly error-prone. It makes a huge number of mutations. So this is part of HIV's strategy. This gives it an enormous survival advantage. So, as you may know, genetic changes are what mutations are what drive evolution. So they allow organisms to adapt to changing conditions, to colonize new habitats, and so on. And so that's what, for example, if you've seen these studies where you have lizards, and if you move them from a territory with black rocks to white rocks, eventually you start getting lizards that change their coat color and so on. The reason is that the ones who have mutations that change their coat color, those reproduce more. So the changes are absolutely random. They're not directed, but the ones who have advantageous changes produce more progeny, and so it drives it into the species. Okay, so from the perspective of you or me, mutations are not that great because, for example, if you think about making a permanent genetic change or a mutation, this can have very bad consequences. For example, one single mutation results in the genetic acquisition of sickle cell anemia. So a single change in the gene that encodes for hemoglobin, which is your oxygen-carrying molecule. Cancers are caused by a gradual accumulation of random mutations in DNA. So for us, this is not a great idea, but keep in mind, we don't reproduce that fast, right? But if you've got something like HIV, where you're going to make millions, billions, trillions of progeny, and the virus, not to anthropomorphize this, but the virus doesn't care if 99.99% are unviable. There'll be some small population where the mutation is advantageous. The rest of it is bad. And so those move on. And so this is the problem. If you have a rapidly mutating virus, you cannot keep up with it with your own immune system, which is not rapidly mutating. So it leads to rare variants that have increased fitness, or they can evade the host immune system even better. Okay, and then HIV has another trick up its sleeve. I said it was a retrovirus. It converts its genetic material, which is RNA, into DNA using reverse transcriptase, but then it does something even more clever for itself. It takes that DNA, and when it's in the nucleus, it integrates a copy of its DNA into the host chromosome. And so a lot of times, this cell isn't even producing these viruses. It's just sitting there. It's doing nothing. But every time it divides, each of its progeny carries the HIV genome so they can produce virus at any time. So this is what happens is that you have latent reservoirs in your body of HIV if you're infected, because there are cells that aren't producing virus, but they've got the ability to do it at any time that they get turned on. So this is why HIV is so hard to eradicate. So as I said, in this country, it's not that bad of a problem for most people, because you can get antiretroviral drugs, which collectively are called highly active antiretroviral therapy, or HAART. You have to give them 3 at a time, usually 3, sometimes 4. If you give a single antiretroviral drug, the virus will mutate to escape from it. So what was figured out, like in the 90s, was that if you give them a combination, so let's say you have all these drugs like reverse transcriptase inhibitors. These are small molecule compounds that are synthesized chemically. They would inhibit this enzyme, reverse transcriptase, or they might inhibit this protease, which is a cleaves proteins, and it's required for the HIV life cycle. So there's all kinds of places that you can target with these drugs. But you can't give just one, ever, because the virus will always mutate to escape it. But if you give 3 or more at a time, the virus can't mutate that fast. So it keeps it at bay, but because of latent reservoirs, a person taking antiretroviral drugs has to stay on it for life. As soon as they get off of it, they have no detectable viral load, so they can't infect other people. But as soon as they get off these drugs, their viral count goes back up. And then, but a bigger problem, I think, is that only 50% or so of infected people in the world are being treated by these drugs, even though they are, in theory, available. So what we'd really like to have is a vaccine, and a vaccine would work if we had one for HIV the way almost all the vaccines you've had in your life work. So what happens is something is injected that's not infectious, but whatever is injected, you raise antibodies to it. And I'll talk about antibodies later, but these are host cell proteins that are specific for a particular thing that they encounter. So the great thing about antibodies is that they can bind to any new pathogen, okay? Okay, so the way it would work is you'd inject something, the immune system would raise antibodies, and then antibodies will clear the infection. So I want to point out that you've probably all heard of or seen antibodies, even if you don't remember them from whatever biology classes you've had, because in how many of you saw Fantastic Voyage? It's a 1966 movie, so most of you would have seen it through, streaming, whatever, of an old movie. So do you remember this scene here? So these scientists are going inside somebody's body where he has a clot or something like that. And so they get miniaturized and they get put in a tiny spaceship. Well, it's not a spaceship, but it gets injected in the person's bloodstream. They're supposed to go out there with these lasers and get rid of the clot because this is a national security issue. And so Raquel Welch right here goes out in a diving suit to go out and get rid of this. And what happens is she gets attacked by antibodies in that movie. So even in 1966, they knew about antibodies, and they depicted them as these like things right here, which they're not, they don't really look like that. I mean, they sort of look like that. But anyway, even back then, they knew about this kind of stuff. And what they had the science right in that if your immune system has never seen Raquel Welch before, it's going to attack her, okay? So that's the point of this. Okay, so if you raise antibodies against HIV, they could actually be neutralizing, which means that some of them, if the right type of antibody would bind to, let's say, the spike right here, and it would interfere with attachment here, so you'd never get it infected. It would never be able to infect. So that's what you want. You want a vaccine that will raise an antibody like that. That's what we really want. Okay, so let's talk more about what antibodies look like. They're sort of, well, T-shaped or Y-shaped proteins, and they have two parts to them. One's called Fab, and that means fragment antigen binding, and that just means the antigen is what is seen by the antibody, and the other part is a constant part that's fragment constant, let's say. Okay, so these are the parts that would bind to the foreign thing, the new thing, and this is pretty much the same from one antibody to another. So the antibodies are going to be specific for different pathogens, which we call antigens, okay? So you have an antibody against cold virus, you have an antibody against HIV, but you have many different antibodies against those two viruses because they could bind to different places on the HIV or on the cold virus, or if you inject sperm whale myoglobin into a mouse, you'll raise sperm whale myoglobin antibodies and so on. So you have all these different antibodies. You have an infinite capacity to make a huge number of antibodies. And if you have an antibody that will neutralize a virus, it will prevent the virus from infecting its target cell, and then you won't have any of these viral progeny. So let's look at what you might be doing. So a typical mammal, that is you or an experimental system that's used in immunology a lot, are mice, but you have the capacity to make at least 10 to the 16th different types of antibodies, probably more than that. So that's an enormous amount. It's actually more than you have lymphocytes. You probably have 10 to the 11th lymphocytes, but you have the ability to make this enormous number of different antibodies. So any new pathogen that comes in, you should be able to make an antibody against it. So I'm going to depict those different types of antibodies with different colors here. Okay, so that just means they're specific for different things. So, okay, and then I defined this already. There are two Fab arms. It means fragment antigen binding, and these are variable, so they're different from one antibody to another. And then there's one FC region, which is pretty much the same, and that is involved in like furthering the immune response. But for the most part, this is the part that differs, and I'm going to show them as these Y-shaped molecules, but this is also accurate because antibodies are flexible and they can move like this, okay? All right, so I want to show you why antibodies don't work very well against HIV. Okay, so here you have, this person is infected with this virus here, and we'll call it a yellow virus. So it turns out, as you'll see, that an infected person is full of many, many, many different strains of HIV. You'll see why in a moment. But only one gets across usually to start an infection. Okay, so this person is infected with the yellow virus right here. And these little things here are the spikes that allow it to infect a new cell. And so when the virus gets in, it gets into host cells and it makes copies of itself. And the copies are slightly different from each other, as illustrated here by these different colors. And so these are random mutations. Now, most of the time, those mutations, the virus is unfit, so it doesn't survive. Or antibodies that are specific for that type of virus come in and neutralize it, so those don't survive either. But some small fraction doesn't get attacked by antibodies and makes fit viruses. So those go on and they make a new set of viruses. And a lot of those are unfit, or they get attacked by antibodies and they go away. But some fraction goes on. A lot of those are unfit or get attacked by antibodies. And so you can see what's happening here is that within a short amount of time, if you're infected with HIV, you get this, what we would call a viral swarm. So inside the infected person, there are many different species of HIV that have genetic changes from each other. And then you've made all these antibodies, which is great. But the problem is that if you look at those antibodies, so what can be done now is you can take HIV-infected donors and you can isolate antibodies from them, and you can characterize them in the lab, and that's being done sort of in many different labs around the world. And most of these antibodies are strain-specific. That is, the blue one would neutralize, say, blue viruses, maybe purple viruses, but not the red viruses. And so they're not useful to the person. They just kill off one set of viruses, but the virus mutates away from those. And so that's what I mean by an arms race, is that it's just this constant thing where your immune system is too slow to make antibodies that could kill off the virus before it changes. Okay, but it is still useful to look at these antibodies, and that's what my lab does. So I'm finally getting around to what we actually do. So if you characterize these and you run tests in the laboratory, you'll find that most antibodies from a HIV-infected person are strain-specific, but there are rare antibodies from not everyone, but rare patients, and those are broadly neutralizing, and we call those BNAbs, or for broadly neutralizing antibodies. So over the years, we and other labs have characterized how those antibodies actually bind to the virus. So this is HIV. It's about 100 or 150 nanometers in diameter, but these little things here, these white things, are the parts that allow it to enter the cell, the spike or the spike trimer, as I've called it, and that's what the antibodies bind to, because that's the only thing on the surface of the virus that is a viral protein. It carries other host proteins, but this is what you have to attack if you're an antibody. Okay, so if you enlarge that spike right there, it's a trimer, meaning there are 3 identical copies of it, and we and others have mapped where those antibodies bind, and they bind to these places that are indicated by colored areas here. Okay, but it's really hard to be a BNAb, or a broadly neutralizing antibody. It's very easy to make an antibody that is strain-specific, but to make one that binds to a lot of different strains is difficult. And the reason is that the spike actually is full of carbohydrates, and carbohydrates are attached by host proteins. And the immune system considers them to be self, meaning there's nothing viral about them. The virus doesn't have the machinery to make carbohydrates or to attach them. And so if you cover yourself in carbohydrates, you're shielding yourself from antibodies. And then there are parts of this that remain the same, but they get hidden at interfaces, and so they're hidden from the antibody, and there's nothing you can do. You can't like force the trimer to fall apart. And what I was talking about here is the so-called glycan shield. So you've got this thing, it mutates very rapidly because all of HIV mutates. The parts that are the same get hidden, and then you have this glycan shield. And I'm going to end with another consideration here, which is there really aren't very many spikes here. And I'll talk about that at the end of the talk. This is actually, if you look at viruses, this is a very unusual looking virus because most viruses have lots of spikes on them, and this has very few. Okay, so I want to say a few general things about how we think about biology and why we do what we do. So it's kind of inspired by this quote from Richard Feynman, who was a physicist at Caltech, where he said, "It is very easy to answer many of these fundamental biological questions. You just look at the thing." So basically, you look at the virus, and you look at the antibody, and you try to figure out what's going on. So to do that, you need techniques like X-ray crystallography, electron microscopy, and so on. So I'm happy to answer questions about how all that works, but I'll just tell you that that's what we do in my lab, is we use various structural biology techniques to look at things, okay? So over the years, we've gotten, these are various structures, and these are computer graphics depictions of those structures. And we are basically chemists, except that to show these pictures, this might have, let's see, 100,000 atoms in one of these. And so we have ways of simplifying them so we can actually look at them. But if we need to, these are three-dimensional structures, and so we can use graphics techniques to turn them around and look at them. We can zero in on where things are. We even have programs to do this, where Anthony West in my lab developed a program where you can look at all of these things on your iPhone, should you want to. That's downloadable from iTunes, but maybe you're not interested in that, but it is available. But anyway, so sometimes we show sort of a space-filling representation where the atoms are enclosed in their van der Waals radii, if you remember that from chemistry. So that shows you how this is an antibody fragment, and that's part of CD4, and that shows you how it would bind. Or sometimes we look at what we call cartoon representations, where the protein has a trace, so it might have 500 residues, and they go from one direction to another, and then we trace that, and that sort of allows you to look for helices and other things like that. Other times we depict them at really low resolution to just look at organization. Or this is from an EM. We put the cartoon diagrams into the density to kind of look at where something fits, or we put some of it in space-filling representation and other parts of it. That's part of an antibody, so to look at where it binds. So we can depict these however we want, and there's a lot of really sophisticated software to interpret the density maps we get into these things. We can color them any way we want. And so the point, what we're trying to do is use insights gained from studying structures to design better broadly neutralizing antibodies. So we get the broadly neutralizing antibody from a patient, and then we try to be bioengineers and make it even better. Or we try to design something we could inject in someone to make a vaccine. So those are two goals in my lab. So I just want to show you, without going into details, this is an example of one of the structures that we did in my lab, where the tan and the pink thing is the HIV envelope trimer, the spike trimer. And all of these are glycans in magenta and blue. And then this green, the green thing and the blue thing are different antibodies. We're just looking at the glycans right now. we're well, the glycans are these spheres right here. We're really interested in places where antibodies could fit in between the glycans. So for example, that red place, we can do varied surface area calculations to address where antibodies might be able to fit and so on. So that's what we do when we get a structure, is we analyze it to figure out, for example, this red place is, we don't want antibodies to go there because this is a non-conserved part of the envelope. So from one strain to another, it's completely different. So what does HIV do? It removes glycans or carbohydrates from that region, so your immune system will target antibodies there. The antibodies neutralize that strain, but then the virus mutates. So it's a useless antibody. So one of the principles we've learned from studying these structures is we have to get rid of these sorts of distracting areas. If we want to make a vaccine, we're going to make antibodies against the wrong parts, and they're going to be strain-specific, and that's not going to be useful. So these are some of the things we're trying to learn. And so I just wanted to show you another movie also, because what I've showed you is the envelope trimer here. And this is, well, it has 3 subunits. The third one is in back, so you can't really see it. And you don't have to care about the details here. What I want to show you is the structure actually changes a huge amount. So people made this is from a review article, you know, and so it's shown schematically. So like you abstract that whole structure I showed you as just this sphere right here. But the point is that after you bind CD4, there's a massive change here. And then that's what allows what I've been calling the membrane fusion. That's the first step in viral infection. And so what I wanted to show you is that, like this cartoon, this is like a schematic with spheres here. Well, we know what that looks like. So this is from a 3-dimensional structure where we know where the atoms are, and we've enclosed them in their van der Waals surfaces, and we have different areas, like this red area right here. We know that that's what this person here is showing as these ellipses, and so we know actually what that looks like. We know where CD4 binds. That's listed as CD4 binding site right here. And we know where the 2 subunits are and so on. And then what we're interested in, well, there's also a structure for that part from 20 years ago. But we're interested in what happens when CD4 binds because, as you see schematically here, there's a massive rearrangement. And so this is from recent work from my lab where a graduate student, I think, yeah. Okay, so we're looking through structural biology. We can actually watch the trimer opening. So you're looking at it from the top, and when it opens, this is what happens. That's yellow is CD4, but when it opens, there's this massive rearrangement due to CD4 binding. So that's closed, and when it binds to the receptor, it opens, and we know a lot of details about the rearrangement that happens, and this is just showing some of those detailed rearrangements. We depict the direction of how strands are going with these arrows right here, and what's being shown is that there's a rearrangement in those arrows, if you saw that, between the closed conformation and then the open conformation. So we're really interested in understanding how that happens, and that's when it's induced by CD4 binding. And then this is now looking down on it, and you can see the red things looking at it from the side. When CD4 binds, that's CD4 right there. The red things move out of the way, and in fact, they become disordered, meaning that we can't really see them anymore. But this is how much this thing moves. So there's a massive rearrangement here. And I should say, this is a computational morph. We have static pictures. We have open structure and we have closed structure. We don't know how it moves between the two, but programs will show that. Okay, so this is really detailed stuff now of what this student, Ji Young, is studying. I think this is too detailed for what you care about, but basically there's this fusion peptide that I talked about, and we can see structural rearrangements in that fusion peptide depending on different conformations that G has been looking at. Okay, and then you know, you can basically see, normally we're not showing them to you, but we can plot, or we can show on this picture all the side chains. That's a phenylalanine right there, and so we know exactly where it's going and how it's moving between different environments and so on. Okay, so supposing we understand all of this, and this is a structure right here, this is an antibody fab, and this is part of the envelope. We could actually either try to make something to inject, which we're trying to do to make a vaccine, or we can just try to make this antibody better so that maybe HIV can't escape from it. So when we started thinking about this in 2010 or something like this, we started wondering why HIV is so much better at evading antibodies than other viruses. And again, this comes from, you know, the answer that we think, this is a hypothesis, but the answer we came to, we arrived at by looking at HIV. So I'm going to bring you through that thought process, and then I'm going to say that we haven't succeeded in making things that counteract this problem, but I want to show you another reason that I think that HIV is such a foe for our own immune system. Okay, so these are a bunch of envelope viruses, and this is what they look like by electron microscopy, and this is just what you'd see in textbooks or virology journals or whatever. And so these are just pictures we got from the literature. Okay, so all of these are envelope viruses, which means that they have a lipid bilayer around them. So they have parts of their life cycle are similar to HIV, but not all of it. But there's a lot of different types of viruses. But anyway, this is one type of influenza virus. It has the lipid bilayer. This is the virus that causes measles. This is respiratory syncytial virus. This is hepatitis B. This is Ebola. Ebola's a tube-shaped thing. But what I want you to think about is that in all of these cases, an antibody response can either clear the infection. So for example, in Ebola, they have purified antibodies they give to people, and they will clear the infection if they get them soon enough. Or you can make a vaccine that prevents the infection in the first place. So the annual flu vaccine you get, the only reason it's not 100% effective is because the flu changes. It doesn't change as much as HIV, but they go around the world, they look for the three or four most common strains in the world. They then make a trivalent or quadrivalent vaccine of those strains, and sometimes they don't predict which one is going to rise up, and then you have breakthrough infections. But anyway, for the most part, there is no vaccine against respiratory syncytial virus, but if an infant gets RSV, it can be fatal, and so they get an antibody to treat it. And so the antibody will clear the infection in an infant. And one thing that's interesting about this is that the envelope spikes are actually presented as a dense array. So if you look at these spikes here, there's lots of them, and they're right next to each other. So do you see that? You can't really see it right here, but all of these faint white things are envelope spikes. And now you look at HIV, and it, to me, looks very odd because it doesn't have very many spikes. And we know in the HIV case, the antibody response is ineffective in that it works, but it's strain-specific for the most part. And so people don't clear HIV infections with their antibody response. And it may or may not be related, but we think it is. The envelope spikes there's hardly any of them, and they're far apart from each other on the most part. Okay, so one thing about antibodies is that, as I said, they're these Y or T-shaped entities, and the parts that they bind the antigen are highlighted here in red. So remember, this is the fragment antigen binding, and the red part is where it would bind the antigen, and it has two arms. Notice it has two arms. And now let's look at influenza. There's influenza again from the last slide. And what's undoubtedly happening, and there's some evidence for this in studies that have been done in laboratories, is that when you have an antibody, it's binding between spikes so that one arm binds here and the other arm binds to an adjacent spike. And so the argument I want to make is that antibodies actually bind best if they can use both of their arms. And the analogy I'm going to make is to what's happening if you or Elmo is trying to hang on to something. So Elmo's like the antibody, and he's trying to hang on to this bar through whatever these things are. These are actually the influenza spikes right here. So in this animation here, Elmo is hanging on, and eventually one arm gets tired, and that's fine, and it falls off. But because he's got both hands, even if there's a mutation here so that the way he's gripping changes, he can still hang on because he's got both arms on it. So the idea is he can remain on the bar even if the handles change. So that's the analogy to mutation, okay? So now I'm going to show you the prediction of what happens to Elmo if he can't reach between these bars and he's hanging on with just one hand. So this would be the analogy to HIV. He's fine as long as, he's got a very good grip here because the bars fit his finger really well. His fingers, I mean. But now if it changes, eventually, he's fine there. But eventually, he's going to fall off because he doesn't have the ability to hang on with both hands, okay? So the idea is, and we published this sort of opinion or prediction, this is a hypothesis. So we called it few and far between, how HIV may be evading antibody avidity. So avidity is the ability to use both arms to bind. And in a physical chemistry sense, what's going on here can be explained by the following. So what you have is you have an affinity of an interaction. So if you remember chemistry, A plus B goes to an AB complex, okay? So there's a certain on rate and off rate for that. And so it's in equilibrium here, so that it's going to fall off every once in a while. But if it falls off and it's binding with two arms, it's still got the other arm there, so it can rebind. Okay, so that's fine. As long as you make high affinity antibodies, you don't need to bind with both arms. But it's when you have rapid mutation, and if you can't bind with both arms, this is what we think is a problem. So here's the idea here. If you've got your normal type of virus, you have all of these antibodies have two identical Fabs, and that permits what we call bivalent binding through between spike cross-linking. So we're differentiating between inter-spike, which means between spike, and intra-spike, which would be on the same spike. Okay, so now what we're talking about is between spikes. So you've got flu, you've got measles, and for the most part, this is what's happening. And even if these spikes change, the antibodies would still work for long enough to clear the infection, let's say. So for HIV, there's very few spikes, and the spikes are far apart. And so most antibodies aren't able to bind with both of their Fabs. And so you've got a problem here as soon as there's a mutation to introduce new strains where the antibody binds more weakly. And so it can't compensate through binding through its second arm. So what we're trying to say is it's HIV's low spike density plus its rapid mutation that is one of the reasons that HIV is such a problem. So how can we, if we're engineers, maybe we can make something with longer arms. And that's not going to work because these are all separated by different distances here. And so there's no common distance, and they don't appear to move very well on their membranes. So this isn't going to work. So what we'd like to be able to do, and we haven't succeeded yet, but we have some leads, is cross-link within a single spike. And if in that case, you'd have intra-spike cross-linking, which would allow bivalent binding, despite the fact that there's hardly any spikes and they're far apart. And so now, if you've got Elmo binding to two places on the same spike, you don't have this problem, even if the spike mutates, then hopefully, so it's mutated now, but Elmo can remain on through these, what we call avidity effects. So that's what we're gearing up towards trying to do. So to summarize what we're trying to do, We're trying to engineer better broadly neutralizing antibodies for both therapy and prevention of HIV infection. And we're also trying to make immunogens for vaccination to actually elicit these broadly neutralizing antibodies. And so with that, this is a subset. I talked about work that spans 10 or more years. I'm only going to show you a few of the people who are presently in my lab. So Harry and Morgan are both doing the vaccine work, which I haven't told you. I've told you very little about. Hao Qing recently got a postdoctoral fellowship at Stanford. He was doing EM. And then Christopher Barnes and G are both doing EM that I told you a little bit about. We get funding from the National Institutes of Health and also the Gates Foundation, and then there are various places at Caltech. So I'd like to thank you for your attention, and if you have questions, please let me know.
So I have the rollable microphone, so you got some questions back there? Thank you. So I have a couple questions, actually. So do you know why more viruses don't have the properties that HIV does, like with fewer spikes and the rapid mutation? Because it seems like it would be evolutionarily advantageous.
It is, except HIV is a terrible virus. It's incredibly wimpy, as that's the technical term. I mean, it's not, but it's estimated like 1 in 250 or something sexual encounters before the virus gets across. This is a terrible thing for actually infecting a cell, to have so few spikes. It is not good. And so if you're a virus like flu, if you think about it, what flu is doing is it's not trying to evade the immune system for very long. It wants to get in, make you really sick, make you sneeze on someone else or on a doorknob, and then it passes on to a new person. What HIV, quote unquote, wants to do is remain in you for 10, 20 years, and then you will infect not that many people, but you will infect enough that there will then be transmission of HIV to get to 35 million people. So it's a successful strategy, but it's not what a cold virus or a measles virus or, it's not what polio would. All of these viruses kind of come in, do things, make a lot of new viruses, and transmit very quickly. So, I mean, I'm kind of justifying. No one knows this for sure. But the thing that I find really interesting is you can engineer forms of simian immunodeficiency virus, which is the non-human primate version of HIV. You can engineer them to have many more spikes, like 150 or something. Okay, so you put those in the lab and you ask how well do they infect? They infect just great. They have more spikes. It's just great. You put them into a non-human primate, and what happens is that virus mutates to reduce its spike number. And it does it very quickly. And that must be because that animal has a functioning immune system. And so it's more, once HIV is in your body, it does not want to have a lot of spikes, I think, to avoid the immune system.
Sort of because it benefits more from being in your body for years and years?
I think so, yeah. That's at least a hand-waving argument.
Hi, thank you. Quick question. So after the antibody binds to the virus, does something clean it up?
Oh, does something clean it up? Yeah, that's a good question. Okay, so there's things called antibody-dependent cytotoxicity, in which cellular cytotoxicity, in which another immune cell comes, recognizes the FC part of the antibody, and then either eats the antibody virus complex or does something else to it. There's a lot of different mechanisms, but it is eventually cleared with that antibody.
Thank you.
I have a question about the larger epidemiological probabilities. It seems like when you clear away all the higher morbidity things, that HIV would be almost inevitable. Is that true in your opinion, or something similar? And in, say, artificial intelligence, if you had enough deliberately injected human pathogens like hackers, eventually it would be inevitable that a similar self-replicating thing would appear. So what is the prior probability that HIV would have started? And secondly, on the posterior side, if you manage to eliminate HIV not through a vaccine, but just driving the population to extinction through lower probability, like Ebola locally anyway, would it just come back?
That's hard to answer, but I can speculate, and this isn't my work. But there's this great book called The Origin of AIDS, and I can't remember the author's name right now, but it came out like 5 years ago. So it turns out that through molecular clock analyses, like you sequence all the RNA from a bunch of HIV viruses and you do a estimation of when they arose through, you know the mutation rate and so on. So you have samples from, the earliest samples I think might be from the 50s, but anyway, there are certainly samples from the 70s. Anyway, you do this molecular clock, and it looks like HIV came out, the current outbreak of viruses, which are called this subset of M viruses, came out in a place near Cameroon in Africa in the 1920s, let's say, okay? And it came from, the speculation is, one of these SIVs, so simian immunodeficiency virus. So there are hunters in that part of the world that hunt, I suppose it's chimpanzees, and when they're hacking them up, they get if they have a cut and the SIV in the blood of the non-human primate gets into them, they got infected. And apparently this happened a lot, like they'd get infected. But SIV didn't, just like once or twice, it mutated such that it would infect a person's cells rather than just the chimpanzee cells. And so now you have this person with HIV, so this hunter, okay? And this probably happened, people speculate, many times. But why did it take off? And the fascinating thing about this book, which has maps and all this kind of stuff, is that there was a program, it was a Belgian doctor who was trying to get rid of sleeping sickness and other things, and he went around to places near where HIV started in Africa, and he had a campaign to inject people with arsenic, which was the only treatment at the time. And you can find YouTube videos of this guy in the 1920s and his helpers, and they go from one villager to another, and they line them up, and there's like 500 of them, and they're using the same needle, okay? And so you can't transmit HIV very easily sexually, but you can transmit it really well through needles, which is why IV drug users transmit it much more easily than you would have it through mucosal transmission. And so apparently there were enough of these campaigns that involved needles and using the same needle that it spread really fast in this part of Africa. And then there were other things going on with blood transfusions and things like that, he has maps of how it spread through these various areas of Africa, and it's exactly predictable with assuming it started, let's say here, it goes to the nearest city. Now you have sex workers that are propagating it. You still have some blood-to-blood transfusion. And eventually, one of the interesting things was it gets to Haiti. So if you remember, Haiti was a place where there were a lot of HIV cases in the 80s, and nobody understood why. Well, it turns out Haitians were coming to the French-speaking parts of Africa as educated doctors and engineers and so on, and enough of them had sexual contact, I guess, with some of the people who were infected that they brought it back to Haiti. But then Haiti had this program of donating blood for money, and that whole blood supply was contaminated. And it got contaminated early on, But then they had very unethical people who paid Haitians to donate vast amounts of their blood under terribly unsanitary conditions, and it spread all over Haiti through that route. It gets to the US and then Europe and so on. And this book is incredible. It's a problem with, nobody did this on purpose. The programs to get rid of sleeping sickness and so on had good intentions, but they didn't understand about transmission problems with blood at the time. So does that
Well, Michael Crichton might say that that's like one specific thing, but is it inevitable that something like that will always happen, like a black swan? One of the things that limits Ebola is people die too soon.
Yes.
But if you kill off anything that makes you die too soon, then it enables at some level, this will be the leading thing that will kill you. And it seems like that's kind of generically hard to prevent.
I think that we can't do the experiment, but if nobody was treated with antiretroviral drugs, it might happen that we eventually as a population would die off and then it would go away or become dormant, like the plague, right? So all these people died of the plague, and rare people who weren't susceptible survived that. But there was a massive death from the plague, right, in Europe and stuff like that. We can't do the experiment because we have interfering things going on with programs to eradicate it. So I mean, I don't know. If the antiretroviral drugs should work, but they're not working for everyone. So I think if we could, with Ebola too, you're right. The reason Ebola is not a problem is people die too fast. That virus is not very good at spreading itself around because it kills its host too fast. But it's a difference in the strategies of the viruses, and HIV is winning. But with Ebola, there is a vaccine. It's just not being used that much. It was developed because Ebola, there aren't that many strains. There are some genetic variations, but not trillions or whatever. So there's been a vaccine for quite a while. I don't know the details of if it's being administered or not, but there is a vaccine. And there's also monoclonal antibody treatment for Ebola.
We have time for just one more question or so. Actually, I think you had your hand up very first, so sorry. There you go.
Thank you very much. From what you have told us, it seems that the arms race, as you called it, between the antibodies production and the virus mutation is the key thing to understand why people get to die from HIV. My question is, could you please briefly tell us about the other side? I mean, how the antibodies are produced by our body.
Oh, okay. Well,
because this is what you haven't told much about, but
No,
it seems to be very important.
Okay, so just to say that's a theory, that's a hypothesis about the arms of the antibody and everything, but how antibodies are produced. So I told you could make 10 to the 16th or so different antibodies. So what happens is you have white blood cells that are made in the bone marrow, and there are precursors to an antibody-producing cell, and an antibody-producing cell is called a B cell. The T cells that are eradicated in HIV have a role in that. They kind of interact with the B cell to help them make antibody. And so you mess up your antibody production a bit in HIV infection. But anyway, so you have this B cell, and it randomly displays a particular type of antibody, let's say a blue one, and the neighbor would make a red one, and another one would make a yellow one, or so on. So now if you get infected with something, or in an experiment, they inject, let's say, sperm whale myoglobin, it goes in and it has all these B cells that have many different randomly displayed antibodies. And it binds to the one that it will bind to, and that one is signaled to make many copies of itself. And so it's called clonal selection. So if you have, say, 10 to the 9th, 10 to the 11th different types of B cells. I'm guessing on the numbers here, but it's somewhere in that range. Then you pick the ones that work, that bind, and you clonally expand those. And while they clonally expand, they actually mutate. So we do this on purpose. We make mutations. So in this case, mutations are good because the mutations make the antibodies bind better. And the B cells that make mutations while they're dividing that make the antibodies bind worse don't get any signaling, so they die off. And so the ones that get better get much better. And then they make antibodies. And some of those B cells go on to make what are called memory B cells. And those can get activated quickly. And that's the basis of vaccines. And so the point is the reason you get sick with the flu is it takes you about 5 to 7 days to make flu-specific antibodies. But because if you don't have the memory B cells against that strain of flu, you have to go through all that. But if you've been vaccinated or you got that strain of flu, a year ago or something, you have these memory B cells, and they can be activated quickly. And so then you can fight off the flu without necessarily getting sick. And so, yeah, there's a whole clonal selection theory, and this kind of stuff is somewhat understood, but there's a lot of research going on into that. So that's kind of a 30-second.
Okay, thank you very much.
All right, well, thank you so much for coming and doing this talk.